Hello everyone and welcome to the new session of the lecture series, and today we'll be discussing the topic cell cycle and cell division. Okay, and we'll do the entire chapter revision today. Alright, and I would like to mention that all the topics covered in this lecture are from the NCERT book. So let's start with the lecture. Okay, let's start with the first topic, which is the cell cycle. So what is cell cycle? The stages through which a cell passes during the cell division is known as the cell cycle. Now cell cycle is further divided into interphase and the M phase. Now M phase is also known as the mitotic phase. Now let's talk about the interphase first. So interphase is further subdivided into G1 phase, S phase and G2 phase. Okay, G1 phase is also known as the gap 1 phase, S phase is known as the synthetic phase and G2 phase is known as the gap 2 phase. So in this diagram is showing you the interphase. These are the stages of the interphase. You can see we have the G1 phase during which cell growth is taking place. Alright. In S phase, known as the synthetic phase, DNA synthesis or replication takes place. And then we have the G2 phase. In this stage, what happens? Final preparation of the cell is going on. So preparation for the mitosis or the cell division is taking place here. Okay. Then after the interphase, till G2 we have the interphase. And then after interphase we have the M phase. Okay, known as the mitotic phase. And during the M phase, what happens? Actual cell division is taking place here. Actual cell division takes place in the M phase. Alright. Now M phase is further subdivided into two. First phase is karyokinesis and then we have the cytokinesis. Now, let's talk about the M phase. Now, M phase, okay, is divided into two phases. First is the karyokinesis, which is known as the division of the nucleus. Nuclear division takes place. And second is the cytokinesis, during which cytoplasmic division takes place. So, we have karyokinesis and we have cytokinesis. Now, let's talk about the interphase first. Okay. Now, interphase is also known as the resting stage. Okay, because the cell is, okay, haven't started dividing, but the cell is called in, okay, or we can say cell is in resting stage. Okay, but the cell is metabolically active and what is happening? There is lot of synthesis and growth is taking place during interphase. Alright. So now we already know that we have the G1 phase first, then we have the S phase, then we have the G2 phase. During G1 phase, what is happening? Cell growth is taking place. Organelles, cytoplasmic organelles like endoplasmic reticulum, Golgi complex, okay, nucleus and all, okay, they divide and duplicate. And there is preparation for the cell division is taking place. Okay, in S phase, what happens? DNA replication is taking place. Okay, and the amount of DNA doubles here. If the amount of DNA here was suppose 2C, here it will become 4C. Alright, if the organism is diploid initially 2N, then 2N contains 2C amount of DNA. Okay, after S phase, here it will be 2C. Okay, but after the S phase, it will become 4C. Alright. So, next again come back to interphase. Interphase is the longest phase. Okay. Here the cell is metabolically active. Okay. It is metabolically active. Okay. It is the longest phase and it comprises about 95%. 95% of the duration of the cycle, cell cycle. Okay. So that's why it is known as the longest phase. Then let's talk about the G1 phase first. So it is the interval between mitosis and the S phase. We have the G1, S and the G2 phase. Okay. So it is the interval between the mitotic phase and the initiation of DNA replication. This is what is G1 phase. Now during this phase, the cell is metabolically active. Okay, there is production of lot of carbohydrates, lipids, as well as RNA and proteins. Okay, so macromolecule synthesis is taking place here. Macromolecules are synthesized. Alright, but there is no DNA replication. There is no change in the DNA content. If the cell was diploid, okay, 2N means, okay, this is the diploid cell, means contain 2 chromosome sets, okay. So the DNA content will be 2C initially. If the cell is haploid, then the DNA content will be 1C, okay. During S phase, what happens? DNA replication is taking place as well as histone synthesis is taking place. Alright. What else is happening? DNA content per cell doubles. Okay. After the S phase, okay, DNA content will double. Okay, due to DNA replication. But what happens? There is no increase in the chromosome number. So this you have to understand. If the organism is diploid, okay, means it contains 2 sets of chromosome number, then the chromosome number will remain same, means it will again remain 2N only, okay, but the DNA content will double. It will become 4C after the synthetic phase due to DNA replication. Alright, so you can say same chromosome number, but the DNA content doubles. Okay. One thing happening again here is there is a duplication of the centrioles. Now again, what is the role of centrioles? Centrioles are involved in organization of microtubules. Organization of microtubules. Okay, and microtubules, what is the role of microtubules? They form spindle fibers. Alright, and where centrioles are present? Centrioles are present in the centrosome. Okay, within centrosome, centrioles are present. 2 barrel-shaped centrioles are present. Centrioles. The next is the G2 phase. Okay, period between the S phase and the mitosis phase. Here what happens? Final preparation for the cell division is taking place. Cytoplasmic organelles, okay, they double. There is synthesis of the tubulin protein. Okay, alright. What is the role of tubulin protein? Tubulin protein is involved in the formation of the spindle apparatus. Alright. So what else is happening? There is a proteins are synthesized and cell growth continues. Okay. This is also happening during the G2 phase. Now let's talk about the G0 state, also known as the quiescent stage, also known as the inactive stage or the phase, whatever. Alright, so what happens? Sometimes some cells such as heart cells, skeletal muscle cells, lymphocyte and some nerve cells, they exit the G1 phase. Okay, they stop dividing, they do not divide, they exit the G1 phase and they enter an inactive stage, okay, known as the quiescent stage of the cell cycle. So what happens if this is the G1 phase? Then we have the S phase, we have the G2 phase. some of the cells, they exit the G1 phase and they enter the G0 or the quiescent phase here. Alright. And what happens during the G0 stage? The cells are metabolically active. Okay. Although it is known as the inactive stage, but these cells are metabolically active, but they are not dividing. They do not proliferate. Okay. They lose the ability to divide. But what happens? Depending on the conditions, the cells can re-enter the G1 phase. Okay. So depending on the condition or depending on the requirement of the organism, what can happen? The G0 cells, okay, can re-enter the G1 phase. Okay, and then they will start dividing and complete the cell cycle. Now, what is the condition of the nucleus after interphase? Okay, interphase nucleus at the end of G2 phase. Alright, so each chromosome exists as a pair of chromatid, okay, joined together by a centromere. So, if this is a chromosome, okay, in the center we have the centromere. This is the centromere and these are the sister chromatids. Okay, so a chromosome consists of a pair of chromatid, okay, joined together by a centromere. The cell is diploid, okay, but the DNA content is double now, means 4C. The centriole is replicated into 2 now, okay. There is a duplication of the centriole. Alright. Now, this was the end of the interphase. Let's start with the M phase now. M phase, okay, known as the mitotic or the meiotic phase. Okay, during meiosis we call it as meiotic phase. So M phase is the actual dividing phase, okay, and have a very short span. Okay, it is the shortest phase. Alright, whereas interphase is the longest phase and comprise about 95% of the duration of the cell cycle. Now M phase is further divided into karyokinesis and cytokinesis. Now let's first talk about the mitosis. So mitosis is known as the equational division. Alright. So first let's discuss about let me first introduce you about this topic, mitosis. So the term mitosis was coined by Walther Fleming in 1882. Alright. Now mitosis usually occur in the diploid cell, or you can say somatic cell or vegetative cell, okay, which are usually diploid, 2N. They contain 2 sets of chromosomes. Alright. So mitosis usually takes place in the diploid cell of animals and plants. Okay, but there are few exceptions such as the haploid male drone of honeybees. Okay, so what haploid male drones of honeybees are? Okay, they contain N, 1 chromosome set. Why? Because they are formed from the unfertilized egg. They are formed from unfertilized egg. Okay, and this process is known as parthenogenesis. Alright, so the male drones are haploid, and how they produce sperms? Okay, they produce these sperms with the help of mitosis. So this is an exception. Usually sperms, okay, which are known as gametes, are produced, okay, by meiosis. But here mitosis is happening in the haploid male drones, which leads to the formation of sperms. Okay, and mitosis is also seen in some haploid cell of the lower plants. Now let's talk about we know that M phase is divided into karyokinesis and cytokinesis. So first let's discuss about the nuclear division, which is karyokinesis. Now karyokinesis is again further divided into these 4 phases. Okay, prophase, metaphase, anaphase and telophase. Now first let's talk about the prophase. So it is the first stage of karyokinesis. And what happens during prophase? There is a condensation of chromatin, you can say, or chromosome taking place. Okay, condensation of chromatin or chromosome takes place. Okay, you can see this is the diagram of a prophase stage. Here what is happening, you can see these are the chromosomes, okay, which are attached at the centromere. Okay, and centromere consists of a region known as kinetochore. Okay, so let's talk about kinetochore now. So if this is a chromosome, okay, kinetochore is a region present in the centromere. Okay, if this is the centromere consists of these two, kinetochore and spindle fiber attached to this kinetochore, which are the regions present in the centromere. Okay, so these two were kinetochore. Okay, now these are the microtubules separating centrosome. Okay, these are the centrosomes, okay, which consist of the centrioles involved in organizing microtubules. Nuclear envelope, you can see here, is disappearing. Okay, so during prophase, what is happening? The cell organelles like endoplasmic reticulum, Golgi complexes, nucleolus, as well as the nuclear envelope, they start disappearing. Alright, chromatin condensation is taking place. Now each chromosome consists of 2 sister chromatids connected by the centromere. Alright, this we have already discussed. Now what else is happening during prophase? Centrosome begins to move towards the opposite poles. This also we have seen. Centrosome, which contains the centriole, begins to move towards the opposite pole, and the centrosome radiates out the microtubule called ester. So if this is a centrosome, okay, it radiates out the microtubules. Okay, these are microtubules which are involved in the formation of spindle fibers. Okay, and this forms an ester. Okay, what is an ester? Ester is a star-shaped structure. Star shape, and what does it comprise of? It comprises of microtubules along with the centrosome. Okay, now the two esters together with the spindle fibers forms the mitotic apparatus. Alright, so 2 esters from the 2 centrosome, okay, and together with the spindle fiber, they form the mitotic apparatus. Now let's move on to the next stage, which is the metaphase. So metaphase, what happens? The chromosome aligning at the equator. Okay, so this is the equator or the equatorial plane. Alright, also known as the metaphase place. So the chromosome, they align at the metaphase place or the equator. And what else is happening here? The condensation of chromosome is completed here. Okay, so that's why we say this is the best stage to view the morphology of the chromosome. Okay, if we have to see we have to study the morphology of the chromosome, then we should study it at the metaphase stage. Okay, this is the best stage. Now, metaphase chromosome is again made up of 2 sister chromatids which are held together by the centromere. Okay, these are two sister chromatids. This is the figure given in your NCERT book. These are two sister chromatids which are held together by a centromere. Now, spindle fiber attached to the kinetochore of the chromosome. This we have already seen. If this is a chromosome, these are kinetochore and spindle fibers are attached here. Now what happens at metaphase? The morphology of the chromosome is most easily studied. Okay, this is the best stage. This we have already discussed. Now let's talk about the anaphase. During anaphase, what happens? First, we have to remember this. During mitosis, in mitosis, what happens? Centromere divides. Okay, there is a division of the centromere and the two chromosomes split and migrate towards the opposite poles. Okay, so what happens during the anaphase? There is a movement of the chromatids. Okay, and not chromosomes. The chromatids move to the opposite poles. Okay, and that is how the number of chromosome number is restored. Okay, chromosomes, sister chromatid you can see are moving to the opposite pole. One is going this way and this one is going the opposite way. So if the organism contains 46 chromosome, centromere will divide and 46 chromatomates will go on one side, whereas 46 chromatids will go on the other side. Okay, so that's why the chromosome number is maintained and it is known as the equational division. Okay, and what is happening here? Genetically similar progeny cells are produced which have same chromosome number as the parent cell. Okay, so the organism is diploid, it will remain diploid. And what else you have to remember? That now the sister chromatid that have separated, they are now called as the chromosome now. Okay, now from now on we will call them chromosome instead of chromatids. The next stage is the telophase. So telophase is also known as the reverse of prophase. Why? Because all the things happening during the telophase are exactly opposite of prophase. So during prophase, what was happening? Chromosome condensation was taking place, but in telophase, chromosome decondensation is taking place. Alright, nuclear envelope, okay, they reappear, okay, and the cytoplasmic also organelles, okay, also reappear. Whereas in prophase, they were disappearing, whereas in telophase, they reappear. That's why it is known as reverse of prophase. In this figure, you can see there is a formation of the nuclear envelope. They reappear. And there is formation of nucleolus also. Okay, during the telophase. Now, what happens? This was the end of karyokinesis. Now, after karyokinesis, we have cytokinesis. Okay. Now, cytokinesis, again, you can say it is of two types, or you can say there are two mechanisms of cytokinesis, how the cell will divide now, or how the cytoplasm will divide. So, in animal cells, what happens? The cytokinesis takes place with the formation of furrow. Okay, and this method is known as the furrow method. So what happens in the furrow method? If this is a animal cell, a furrow appears here in the plasma membrane. There is appearance of furrow in the plasma membrane, and what does this furrow then move inwards? Okay, and what does it do? It moves inward and finally it divides the cell into two. Okay, so there is an appearance of a furrow initially in the plasma membrane. This furrow gradually deepens, means move towards the inwards, okay, and ultimately joins in the center and this divide the cell cytoplasm into two, thus forming two cells. So this is what is happening during the animal cell. But in plant cell, what happens? Okay, in plant cell, a cell plate is formed in the center. Okay, if this is a plant cell, a cell plate is formed in the center. In animals, the furrow was formed in the plasma membrane, and there was a growth inwards. Now, in plant cell, what happens? A cell plate is formed in the center, and the cell plate now move outwards, okay, towards the lateral walls. So in plants, this mechanism is known as cell plate formation, okay. So what happens? A wall formation starts with the appearance of a cell plate in the center of the cell, okay. So this cell plate is also known as middle lamella. Okay, and what is the role of middle lamella? Middle lamella joins the primary wall of the two cells. Okay, so a cell plate is formed in the center and it grows outwards, okay, and meet the existing lateral walls. So this is the cell plate, okay. This is growing, okay. The vesicles which contain the cell wall material, okay, fuse with the cell wall. It continues to grow and finally meets the lateral walls, okay, and divides the cell into two. So this is what is happening during the cell plate formation in the plant cell. Okay, now let's move on with the next syncytium. So next is the syncytium. Syncytium, what happens? It is a multinucleate condition, okay, that arises in some organism when karyokinesis is not followed by cytokinesis. Means there is a nuclear division, but there is no cytoplasmic division. Alright, so what will happen? If a cell, there is a nuclear division, nucleus will divide, but this nucleus will remain in the same cell. Alright, there is no cytoplasmic division. Alright, example can be liquid endosperm present in the coconut. Now let's talk about the significance of mitosis. Okay, why mitosis is important? First is mitosis contribute to the growth and the repair of the organism. Alright, how it contributes to the growth? Okay, we know that the apical and the lateral cambium. Okay, so growth occurs with the help of the apical and the lateral cambium. Apical cambium is present in the, you can say, root apical meristem. There is one root apical meristem, and then we have the shoot apical meristem. So the cells in these meristem, they divide with the help of mitosis and contribute to the growth, whereas lateral cambium, okay, such as the vascular cambium, okay, example can be vascular cambium or core cambium, okay, another example. So lateral cambium helps in the, okay, increase in the width of the organ or the organism. Alright, so cells in these regions, they divide with the help of mitosis and contribute to the growth. Okay, mitosis also contribute to the repair of the cells. Okay, for example, upper layer of the epidermis, cells in the upper layer of epidermis, whereas cells of the lining of the gut are also repaired with the help of mitosis, and some blood cells, okay, they are also repaired with the help of mitosis. Alright, Now, mitosis is the second point is mitosis is the basis of the asexual reproduction. Alright, like in prokaryotes. Okay, bacteria. Okay, asexual reproduction takes place with the help of mitosis. Alright. Now, mitosis also helps in vegetative reproduction. So, vegetative reproduction is a type of asexual reproduction. Okay. Now, with the help of mitosis, chromosome number, okay, as well as the genetic constitution of the progeny cells remain the same. Alright, this we have already discussed. If the organism is diploid, it will remain diploid. Okay, and after mitosis, two genetically similar offsprings, or you can say the progeny cells are produced. Genetically similar progeny cells are produced with the same chromosome number. Chromosome number will be same. Okay. So here, but this I have written that the daughter cells produced or the progeny cells are produced are qualitatively and quantitatively similar. Okay, means same chromosome number and same genetic constitution. Alright. So this was about the mitosis. We will discuss about the meiosis in the next lecture. Thank you very much for listening.